Yeah, just keep your distance a bit. Okay, shall we continue, guys? There's an announcement before we start. You want to use this mic?
Yeah, that's true.
Okay, go ahead.
Hi, everyone. Hi, everyone. Before you proceed with your class today, I just wanted to quickly share some exciting upcoming events hosted by BIOSA this month. My name is Khaled. I'm the senior representative for BIOSA. And first up, we have our CV and cold emailing workshop panel happening on Friday, November 14th from 5 to 8 p.m. If you're looking to apply for research positions or lab roles, this event is perfect for you. You'll learn tips on how to improve your resume, how to cold email professors, and where to apply for research opportunities. Next, I'm very excited about the BIOSA's Professional Development Night on Monday, November 18th from 6 to 8. This is actually an event that I organize with my fellow senior representative, Zainab. And this one's all about helping you build career and communication skills in science and research through interactive workshops led by grad students and industry professionals. We'll have sessions on mock interviews, public speaking and research, and networking, with obviously free snacks and networking mixer at the end. Then, on Wednesday, November 19th from 5:30 to 7:30, we're hosting a UE Chen research panel. This is a chance for you to hear directly from 5 principal investigators from the University Health Network, whose research span cancer biology, neuroscience, nanotechnology, and transfusion medicine. You'll learn about their work and how you can get involved in research as an undergrad. And finally, also mark your calendars for the Medical and Nursing School Affair on Wednesday, November 27th from 12 to 3 p.m. in the meeting place. You'll be able to chat one-on-one with admissions officers and student representatives from programs like UFT Medicine, UFT Nursing, McMaster, and NOSM. It's a great chance to ask questions about requirements, the MCAT, and the application process. So definitely check it out if you're planning to apply to professional school. That's everything coming up this month. If you missed anything now or want to register for any of these events, you can check out the Instagram page at UTSC BioCell and check the link in our bio for all the details. I hope to see you at these events. Thank you so much. Thank you.
All right, continuing on. All right, so we saw how the DNA for this virus gets replicated, or these viruses get replicated. But keep in mind that your intermediate and the late genes are post-replicative genes. So what that means is they are only transcribed after DNA replication has started. So you don't get replication of these or expression of these genes until the DNA replication has started. It uses various promoters and initiation factors, just like you'll see for eukaryotic cells. And if to initiate the intermediate genes, it requires your intermediate transcription factors, okay? In addition to that, you also need cellular factors that are involved in mRNA metabolism as well. Now, for the late genes, you require your late transcription factors. And it may require early products as well as a cellular factor. So early gene product as well as a cellular factor that may be required for these genes. Now, after replication, when you start making mRNA, one thing that happens here is, again, you have a poly A tail. So poly A gets added to the mRNA. And the 3' end is actually very heterogeneous. Okay? So it's not identical. There's some variability in the 3' end of this RNA. Because there's a slippage mechanism like we saw earlier, where the polymerase can stutter, for example, and that adds the poly A heads in this case. So you have a slippage mechanism that adds these poly A heads, and the heterogeneous ends from or the 3' ends, they result from inefficient transcription. Okay? Because what ends up happening is the transcription doesn't terminate at the right site, so it's very inefficient that way. But what you see is transcription terminating at multiple sites, and as a result, you have this huge variability, or this variability, not huge, but there's a variability on the 3' end of these RNAs. Now, when you synthesize all this and you replicate your genome and you made all your proteins, you can start to assemble the virus. But the assembly of these viruses or these virions is actually a very complex process. Because what ends up happening is it takes over the host machinery. So you have your ER-derived vesicles. So you're making new vesicles. So you synthesize these rigid crescent-shaped structures in the cell, okay? And these are the ones that will become the mature viral envelope. Thing is, this is not connected to any pre-existing internal membrane structure. So this is still a question mark. We don't fully know. But the way it works is you have your ER-derived vesicles and you have your scaffolding proteins. They will attach and form this crescent-shaped membrane structures. So you have these membranes with scaffold proteins. And from there on, these scaffolds or these crescents will basically combine together to give you your new virion or nascent virion. Okay? And within that, the core proteins will get packaged, the DNA will get packaged, and then you form your immature virion. So you seal this whole particle. And again, your lipids are delivered to these crescents from the ER vesicles. And these ER vesicles, they actually have viral envelope proteins. So you synthesize those proteins in the ER, and then from there on, they get packaged into these vesicles that combine together to give us our immature virion, which then matures into a mature virion. So what we have here is this kind of area here. There you have your DNA replicate. DNA has been replicated. You're expressing multiple genes, and you're carrying out your synthesis. And from there on, you start packaging this genome into these membrane structures. So you get your immature virion. So this is the nucleotide structure that we talked about earlier. Then it gets to mature virion. Okay? Everyone clear on that? Yes? Okay. And these crescents, like I said, they tend to mature into spheres, so spherical structures. So these spherical structures, they enclose the viroplasm, like we saw earlier. And again, this viroplasm is fairly electron dense, and it has the viral core proteins, as we saw in that electron microscope image. Okay? And once the DNA enters, it forms these discrete and dense cores of protein and nucleic acid. So these are, like I said, these are called the nucleoids. Now, the immature virions, by the time they become the mature virions, they have to develop an internal core as well as those lateral bodies that we saw earlier in the structure. This is done by rearrangement of the materials that get captured as these spheres are basically closing. Okay? So whatever material that gets captured as you form these structures will be used to make these lateral bodies. Okay? And when you do this, the virus acquires its final shape and you get your mature virion. And most of these new virions, they remain as mature virions inside the cell. So intracellular mature virions. A small part of them, a small proportion of them, they undergo further maturation and they become the extracellular virus or extracellular virion. And this is designed for export and cell-to-cell spread of this virus. Okay? So here's your MV. It's going to acquire more membranes, and from there it will exit the cell. And this will then go and bind other cells. So you'll have your cell-to-cell spread. Because what ends up happening is your mature virions actually can get wrapped in Golgi-derived cisternae. Again, Golgi-derived membrane structures. This adds an additional lipid layer as well as viral proteins to that surface. So remember, proteins get glycosylated very heavily in Golgi. So it makes sense that the membrane derived from that will have these viral proteins as well. So this makes what's called the wrapped virus, or WE. Okay? These wrapped viruses, they also have two bilayer membranes, and they are transported to the plasma membrane through their actin tails. Okay? So if you see all these little red worms, that's basically the actin tail. And what I want you guys to actually pay attention to is the green dots at the head of those tails. Okay? So let's look at this one here, this area here, because that's very clear. See your tail, then you have your little green dot right at the top of the tail, or right at the end of that tail. That green dot is the virus. Okay? So in this case, the vaccinia virus is green, and this can actually move about in the cell by forming these actin tails. Okay? So it modifies the host cell actin, and it can actually move from one place in the cell to another. Okay? And again, the outermost membrane will fuse with the cell membrane. So from this wrapped virus, it will start to fuse with the cell membrane, and it will release the extracellular virus particle. Now, one thing to add on is that these extracellular viruses can actually remain attached to the cell membrane through things like microvilli. And you can see that here. You see these little projections coming off the cell, and from there you have these green dots on the end. So this virus, even after being in the extracellular environment, is still attached to those microvilli in the cell, or that are on the surface. Okay? And some of the other viruses, along with vaccinia virus, they actually produce these actin comet tails. You can see that with Ebola virus as well. You can see that with baculovirus, and you can see that with vaccinia virus. Basically, they promote actin polymerization and depolymerization. And this is a very common mechanism used by pathogens to actually move about in the cell. Okay? So they affect a signaling cascade that controls the actin cytoskeleton remodeling within the cell. Okay? And these viruses then use this actin tail as propulsion, and then also for budding off the host cell. So let's look at a video, and that will make a bit more sense. So in this case, it's a vaccinia virus that's infecting a cell. And what I want you guys to notice is these little tails and how these viruses move about in the cell. So you can see the virus actually moves around in the whole cell. It's like little wiggle worms. Okay? All those little things are actin tails, and they have the virus attached on one end. So these viruses are basically hijacking the actin polymerization and depolymerization within the cell and using that or exploiting that to move about in the cell and also to come outside of the cell. Okay? All right. In addition to all this, remember our cells have a defense mechanism against these viruses. But pox viruses, they make several different proteins that actually target the host defense mechanisms. Okay? So again, remember our cells have defenses against pathogens. But there are proteins being synthesized by this virus, these pox viruses. So these are the different genes. And this is the system that it infects. So you can see that is infecting a lot of immune system molecules. So interleukin, interferon, chemokines, complement receptors, TNF, which is a transcription factor. Okay? And you can see how it inhibits or what it does with that particular system. Okay, again, don't memorize this. Just understand that pox viruses do interfere with the host cell defense systems. All right, so here's the life cycle of the pox virus. So if you can explain all this, then that means you understand the whole process. Okay? Everyone clear? Any questions so far? Anyone? Yeah. Sorry? So it's the vesicles as well as the mature viruses that can move around using the actin tails, yeah. Okay, you can see this here as well. Okay? All right, if there are no questions, then we'll continue. Next thing we're going to look at is papillomaviruses. Okay? This comes from the Latin word papilla, which means nipple, and a Greek word oma, which is tumor. So this is basically referring to warts, okay, or papillomas. Okay? This virus has a naked icosahedral capsid. So it's basically, this is the electron microscope image of HPV. This is the model, and this is how the virus is actually organized. Okay? It has a circular double-stranded DNA genome. So this is again something unique that we are seeing today. It's a circular double-stranded DNA genome. About 8 kilobases, so much smaller than vaccinia virus. And DNA is actually packaged as a mini chromosome within this virus. So it basically takes cellular histones that make up the nucleosome, and you have similar kind of organization here. So you have your genomic DNA that wraps around these cellular histones. And that's present in the core of the particle, and then you have your capsid proteins on the outside. Okay? So in this case, T is 7. So you guys can calculate how many subunits you'll actually need. So this is a schematic, and like I said, your DNA is packaged as a mini chromosome. Okay? Now there are multiple different types of HPVs or papillomaviruses, and they have hosts that include cattle, dogs, deer, so lots of vertebrates, and there are some oncogenic human papillomaviruses. Oncogenic means they cause tumors, okay? And this is a major cause of genital tract cancers, and they can cause things like genital warts, so condylomas, okay? It can cause neoplasias, which is uncontrolled growth of the cells. And finally, the invasive squamous cell carcinoma, okay? So again, you can see that these various papillomaviruses, they have different sites of infection. So multiple different types, they are just numbered, HPV 1, 2, 3, and so on. And you can see where you will see these infections. So things like hands, oral cavity. These three are the most serious ones because they infect the genital tract and they can cause things like cervical cancer. Okay? So this is what we will look at in a bit. Now, when we look at the disease with these viruses, they cause benign warts at specific sites. So in the skin, mucosa, even in the larynx. But that depends on the viral strain. Also, these viruses, they get transmitted with direct contact. Okay, so you have to come into contact with those tissues, okay, or with the virus particles. Some types, they cause the cervical carcinoma. This is a sexually transmitted disease. It's a major cause of cancer in women. Okay, so the most common is the HPV 16, 18, and 31. Okay, that's what I highlighted in that previous table. They can also cause other anogenital cancers, so cancers of anus as well as genital organs in both men and women. Okay, it can also cause cancers of the oral cavity, oropharynx, as well as tonsils. So oropharynx is this area right here, and it can actually cause cancer in this area. Okay? Now, like I said, the genome itself is circular, is double-stranded DNA, and has about 8 to 10 open reading frames. Okay? There are 6 early genes. They are termed E1, E2, E4, E5, E6, and E7. What's strange here is there is no E3. So we go E1, E2, and then we go to E4. I don't know why that's the case, but apparently that's how it was numbered. Okay? There are 2 late genes as well. So you have your early genes as well as late genes, L1, L2. And only one of the DNA strands is actually used for mRNA transcription. So only one strand will actually be used for making RNA. And if you look at the genome itself, this is the circular genome. You have your various genes, your E1 to E7, and then your late genes here. And what you'll see is that they actually have early promoters and late promoters. There's also a poly-A tail that's added to the early transcripts as well as to the late transcripts. Okay? Now, this is just a linear representation. Just keep in mind that this actually exists as a circular molecule. So if you were to look at this, this is how the genome is organized. You have your E6, E7, E1, E2, E5, E4, and then you have your L2 and L1. And in addition to that, you have the control region, which is the LCR right here. Okay? Everyone clear? It's a fairly complex virus, so make sure you guys understand how it's organized. Now, the viral mRNA, it gets made from 2 promoters and 2 polyadenylation signals. There's an early promoter that's located upstream of your E6 gene, so just upstream towards the 5' end of the E6. And you have a late promoter that's upstream of your E1 gene, which is right here. So here's the early promoter, and here's the late promoter. And there are 2 poly-A signals to add the poly-A tail. You have an early one, and you have a late one. Okay? And these both are downstream of your E5. Okay? And if you want to see the function of these genes, you can look at this table. So for example, your E1 is a helicase. Okay? And this is what will initiate DNA replication. What do helicases do? just based on name, what can you figure out about the enzyme? Yeah. It's going to unwind the double helix molecule. So it's necessary for initiating DNA replication because this is the protein that's going to bind to the origin of replication. Okay, then you have all your other different proteins. You have your L1 and L2 are basically your structural proteins. Okay, so capsid protein, they're both basically capsid proteins that will encapsulate your viral DNA. Okay. Now, if we look at the entry of how HPV actually enters the host cell, it's actually very complicated. It's not an easy mechanism. Okay? There's multiple different molecules involved, and it will bind to the target, and from there on, it gets internalized in the cell. So it could be dependent on multiple different mechanisms. I'm not going to go into the detail, and I don't expect you guys to know the details as well, because there's quite a lot of proteins, and there's, for example, conformation changes that will change the affinity for the receptors on the cell surface. So for example, if you have a conformation change here, it's going to decrease the affinity to the primary receptors. Your membrane, again, actin cytoskeleton, is remodeled after the signal goes in. Your membrane invaginates, and it's going to be endocytosed. It can be endocytosed in clathrin-coated pits, through lipid raft domains in the membrane, or even independent endocytosis. Okay? So you can see all the different proteins and how this virus will get in. Okay? But the mechanism itself of how it works is very interesting. Okay? Because the infectious cycle of this virus, it actually follows the differentiation of epithelial cells. So it needs to get to the basal layer of cells first, where the stem cells are. Okay, it's going to go and infect those stem cells. Okay? So the virus will infect the mucosal epithelium, and it will have to reach the non-differentiated basal cells, as you can see right here. Okay, so this is the basal layer. So remember, then for epithelial cells, for epithelium, we have a basal layer, we have a spinous layer, a granular layer, and finally your keratinocytes. Okay, so these are basically dead cells at the surface. But inside, you have a reservoir of stem cells where the cells are dividing and differentiating and moving up through that epithelium. Okay? So cells will divide here, and as they divide, they will push these cells up. These cells are dead cells. They are rich in keratin, for example, and these can get removed from our skin, for example, or from the epithelium. So we need a constant replacement of these cells. So how does that work? You basically supply that from the bottom, from the basal layer. So what the virus does is actually gets to the stem cells at the basal layer, and that's what it infects. Okay? Because it has to reach these non-differentiated cells, and then it attaches to the proteoglycans, so heparin sulfate proteoglycans. It binds to integrins, so alpha-6 integrins, and then it enters the cell through endocytosis. Now, in this case, the viral DNA gets inside the nucleus, so it's maintained in the nucleus. It replicates modestly until it reaches the right kind of differentiation spot. Okay? So this viral DNA will get maintained in the nucleus. It will replicate modestly whenever the DNA is replicated within the cell, and it will stay like that until the cell differentiates into a keratinocyte. Okay? So here's your stem cells getting infected, your DNA getting replicated, so your viral DNA is replicated, you get your early gene expression, and as the cells differentiate to become keratinocytes, you're going to start synthesizing your viral proteins, etc., and assemble the virions. And once on the other end, the virions will be released from these cells. Okay? So very interesting mechanism where it has to infect the stem cells, and as the cells mature, the virus becomes more active. Okay? And eventually, it will be released into the surrounding area. Your E1 and E2 proteins, they bind to the origin of replication. So if you were to, this is again another representation of the genome. So what you'll see is this origin of replication is actually present in the LCR region, okay, the long control region. So if you look at if you were to zoom in on this long control region, which is here, this is where the origin of replication is. You can see that as ORI, okay? This LCR region is also sometimes called the upstream regulatory region, or URR. It has enhancer sequences that are specific to keratinocytes. So it has enhancer sequences that may be specific to your keratinocytes, and as a result, that's where the gene expression will happen the best, okay? So again, if you were to zoom in on this LCR, this is how it's organized. You have an enhancer region, and you have your late regulatory sequences as well, okay? Your late promoter, so remember we talked about the early promoter and the late promoter. The late promoter is actually active in differentiated keratinocytes. It's not active in undifferentiated cells, okay? Some of these transcripts also get polyadenylated at the late site. So remember we saw the poly A signals that are early and late. So some of these transcripts will get polyadenylated at the late sites. And that produces lots and lots of E1 and E2 proteins. And once you increase the level of these two proteins, it results in increased DNA replication, and you produce thousands of DNA molecules per cell. So again, here's your uninfected epithelium, here's your basal layer, and if we look at the HPV-infected epithelium, what we see is your early gene expression happens in these cells, and from there on, it will continue to increase. Late gene expression happens somewhere here, and that's where you have productive replication. And finally, once you get to the higher layers, your virion assembly will continue, and you will start to release the virions into the surrounding area. Now, these virions are obviously free to go and infect other cells. Your E1 protein, like we saw in the table, is a DNA helicase, okay? It locally unwinds the DNA. So in that area, it's going to unwind the DNA double helix. Your E2, it will bind to a specific site near the origin of replication, and you form this E1, E2 complex, okay? And what this does is basically it increases the affinity of E1 for the origin of replication. So it will bind more strongly to that origin of replication. So here you can see that E1, E2 complex. This is the DNA binding domain of these proteins. So you have your E1, E2. TAD is transcription activation domain, and HD, I think, is the, I can't remember off my head. HD, I think, is the binding domain here, like the protein binding domain. All right, so these proteins, they basically use ATP, and you form this double trimer that will allow you to unwind the target genomic DNA, okay? You also have your topoisomerase, which, what do topoisomerases do? Anyone remember from B11? So it loosens the supercoil, okay? So remember, it's a circular genome. So what happens if you have a double-stranded helix and you try to open this? What's going to happen to the rest of the molecule? Yeah. Okay, so what's going to end up happening is you have a circular molecule. Let's see if I can do this a little demo here. So let's say this is my circular molecule. Okay, it's a double-stranded DNA helix. Can you guys see that or no? At the back? I don't want to break the phone. Okay, let's try it with this wire. All right, so here's your circular molecule. Can you guys see? Yes? So if I were to take one end of the double helix and I open it, what's going to happen? The helix is going to turn, yes? And as it turns, what happens to my wire? If I were to turn this, it becomes tangled. Okay? So topoisomerases, they nick one strand so the DNA doesn't become tangled like that. Okay? Because once it becomes tangled, the replication will stop. The proteins will not be able to go through and carry out their function. Okay? They will stall. So topoisomerases are involved in this, and you have your DNA primase that adds the primer, then you have your DNA polymerase that replicates the DNA. Okay? All right. Now, what we also see with this is that there are a number of different cellular transcription factors and other regulatory molecules that bind to these regulatory regions in the DNA for transcription. Okay? Now, continuing on, your E7 protein is a fairly small protein, so roughly about 100 amino acids. And this is a protein that interacts with the cell cycle regulatory proteins. Particularly, it interacts with the retinoblastoma or RB protein. Okay? So again, this RB protein or retinoblastoma protein is a major protein in the cell. It's a tumor suppressor protein. Okay? It represses the activity of specific class of transcription factors called the E2F transcription factors. Okay? Now, if this, as you can imagine, if this protein, because it's involved in cell cycle, if this protein is affected, you tend to develop things like cancers. Okay? Because now you're messing with the cell cycle. Okay? So what ends up happening is, normally, retinoblastoma protein will bind to your E2F transcription factors. Okay? Now, this can lead to cell cycle arrest. So the cell doesn't really replicate, so it blocks the transcription of cell cycle genes. But your E7 protein actually binds to the retinoblastoma protein. And as a result, it forms a complex. But now, your retinoblastoma protein cannot bind to the transcription factor. So it cannot bind to E2F, and as a result, it activates your transcription of cell cycle genes. Okay? So you're not inhibiting the cell cycle genes, and as a result, the cell cycle just progresses. Regardless of what's happening in the cell, whether it's time to divide or not, the cell cycle will continue. Okay? Because normally, this retinoblastoma protein gets phosphorylated by a CDK cyclin complex. CDK cyclin complex, or the expression of, I should say, the CDKs and cyclins is highly tightly controlled in the cell. Because cyclins, they are involved in basically cell cycle where the cell can actually start to divide. Okay? So that's why they're called cyclin, because their concentrations go up and down very predictably depending on the cell cycle. Okay? So these cyclins, or CDK is a kinase. It complexes with the right kind of cyclin. and it will phosphorylate the retinoblastoma protein. So when the right signal comes in and the cell is ready to divide, this is the normal process. It phosphorylates retinoblastoma protein, E2F becomes free, and the cell cycle progresses. But the virus itself is now making a protein that's messing with the cell cycle. So you can imagine the results of this process. Your E7 can also actually trigger ubiquitin-mediated degradation of the retinoblastoma protein. Okay? So like I said, it's a tumor suppressor protein. It represses the activity of your E2F transcription factor under normal conditions. Okay? It binds to E2F and blocks the activation of cell cycle genes that are involved in going from G1 phase of the cell cycle to the S phase, or the DNA synthesis phase. Okay? Your CDK cycling complex phosphorylates retinoblastoma protein, results in dissociation from E2F, and as a result, you start expressing cell cycle genes. And one of the factors that blocks this is the P53 pathway. So P53 activates the transcription of P21, which blocks the phosphorylation of this retinoblastoma protein. Okay? And like I said, your E7 protein is a small protein. It binds to cell cycle proteins, so particularly retinoblastoma protein, and it can lead to degradation. Okay? Eventually, what you get is, because the cell cycle has not been controlled at this point, you get replication of viral DNA to very high copy numbers. So basically, you replicate this DNA multiple times, and you get lots and lots of copies of that DNA. And this obviously is not normal for the cell, and it leads to things like apoptosis. So P53 can trigger apoptosis of these cells. But again, when you have lots and lots of cell cycles going on, so increased cell cycle progression also leads to increase in P53, and eventually the cell will undergo apoptosis. Okay? But there's a E6 protein, the viral E6 protein, that actually controls the level of your P53 protein. Okay? So now the virus is messing with one of the major cell cycle regulators, which is P53. Okay? So you inhibit this or control the levels of this protein, and this allows the virus a sufficient time to replicate. Okay? So basically what the virus is doing is it's interfering with the normal cell cycle to give itself more time to replicate its genome. Your E6 associates with a ubiquitin protein ligase, also sometimes called the E3A, okay? And this complex then triggers the ubiquitination of P53. Once you ubiquitinate proteins, what happens? They are degraded by proteasomes. So here's your P53, here's your E7, and P53 can be triggered by things like DNA damage, etc. It can trigger synthesis of P21 or BAX. This will inhibit the cell cycle. And BAX is one of the key proteins that's involved in apoptosis as well. Okay? But your E6 bound with E6AP will degrade your P53. And as a result, this doesn't happen. Your apoptosis won't take place. Okay? Now, if you take E6 from the non-oncogenic HPVs, it does not mediate the degradation, okay, of your P53. It also, your E6 also interacts with a number of different cellular proteins that are involved in things like signal transduction, DNA replication, apoptosis, and as well as calcium metabolism in the cell. Because remember, calcium is a very strong signaling molecule. Okay? So you mess with the calcium metabolism, you're messing with the signaling within the cell itself. Okay? And if you inhibit the retinoblastoma protein, it leads to uncontrolled proliferation of the infected cells, as well as you lose the ability of P53 to induce apoptosis. So this is a very potent oncogenic combination, okay? Because there's some synergism between your E6 and E7, as well as someone's predisposition to cancer. So if you have, for example, a defective copy of P53 to begin with, this is going to make it worse, okay? And for example, the cervical cell lines, like HeLa cells, they have actually copies of part of the HPV genome integrated at different sites within the cellular genome. And this integration happens only in cancer cells. You do not see this integration in non-cancer cells, okay? So if a HPV infects, chances are you're going to form those papillomas or wart-like structures. It may not lead to cancer, but in some cases, when it does lead to cancer, we see that part of the genome actually integrates at different sites within the cellular genome. And one interesting thing that happens is the integration actually results in the loss of all your viral genes except E6 and E7. And as a result, you get the expression of E6 and E7 proteins and mRNA within the cell. So far, the evidence actually indicates that E7's main function is to induce cell proliferation, and that leads to formation of tumors. Now, E6 actually has a cooperative role in increasing the size of the tumors, and it can actually result in progression of malignancy, okay? So this is a good target. If you were to inhibit these gene products, that's a potential target for therapeutics in these kind of cancers, so cervical cancers. All right, I think we'll stop here. I know there's only a couple of slides left, but I think we'll stop. Let me see. Give me a second. All right. Let's just finish it because there's only 2 slides, and we do have 3 minutes left. All right. So after you've done your protein expression, etc., you start expressing your late proteins, so your L1 and L2. So this is what promotes the encapsulation of your newly replicated genomes. As a result, your virions are now released from the uppermost layer of the epithelium. So think about how neat this mechanism is. It goes to the basal cells, infects the stem cells, and from there on, it will start maturing as the cells mature, okay? And finally, when you get to the surface, to the uppermost layer, that's where the virus will actually get released. The viral capsid is actually formed from these capsomeres, okay? These are, again, protein subunits that make up the capsid. And they are present as pentamers of your L1. So here's your L1 monomers. You take 5 of these, you form these pentamers. You take 72 of these capsomers, and you form this kind of structure, okay? Your L2 protein, it actually enhances the formation of the capsid. So L2 protein gets in the middle of these pentamers, as you can see here, these red dots, okay? And there's some evidence that it's involved in intracellular trafficking of the virus. So it deposits the viral DNA into the nucleus. So here's your double-stranded DNA genome. You make your capsomeres as well as your monomers, and they will form this virus particle which can be released from the cell through lysis. Okay? Now, you can actually make vaccines by assembling this L1 into this virus-like particles or VLPs. These VLPs do not have a viral genome, so they cannot really replicate inside the cells. So you can use this as vaccine. That's what some of the HPV vaccines are. They use these virus-like particles to induce immunity. Okay? And for papillomaviruses, if you look at diagnosis and treatment, basically to diagnose this, let's say for cervical cancer, there's a Pap test that recognizes the morphologically abnormal cells, as well as it's an effective mean of reducing cervical cancer deaths, okay? Because the early detection actually helps prevent these deaths. So this whole procedure, it allows for identification of the morphologically abnormal cells, and your cervical warts and precancer cells are routinely removed in different procedures. So this prevents development of cancer. And this is often combined with DNA tests for presence of HPV. So you can actually test for the presence of HPV. Now, there are some obstacles. So there are, for example, lack of trained pathologists in some underdeveloped countries, as well as cultural restrictions, because Pap test is a very invasive procedure, okay? So there are some tests that have been developed which are self-sampling DNA tests. So this is something where it's headed, where people are developing these easy tests. There are vaccines as well. So Gardasil, Cervarix are the vaccines used for HPV. These are commonly available, and they're part of free vaccination in several countries. So typical vaccination age is about 11 to 13 years old, but it can be given to older individuals. But in older individuals, you need a slightly higher dose, okay? All right, so here's your cycle, the virus replication cycle. So make sure you guys can explain all this, okay? Next week, we're going to be looking at vaccines and antivirals, and that's more or less is our last lecture next week, okay? We're going to talk about vaccines as well as antivirals, and that's it. You're done after that, okay? All right. I'll see you guys next week.